Okay, well, I'm very pleased to be able to introduce Dr. Balwant Tuana as our next speaker. Dr. Tuana is a professor of pharmacology in the Department of Cellular and Molecular Medicine at the University of Toronto. He's the principal investigator and co-lead of the Heart Failure Cluster at the Ottawa Heart Institute. He did his graduate training at the University of Manitoba and a postdoctoral fellowship at the University of Toronto. He is a scholar and a career investigator of the Canadian Heart and Stroke Foundation. Dr. Tuana's research has led to the identification of several novel genes in the human genome, which encode important proteins that are impacting various areas, including development, as well as muscle, neuronal, and cardiovascular function. The title of his talk today is Cell Cycle Dynamics in the Growth and Death of Myocardium. And we look forward to hearing his talk. But before then, I'd like to present you with this plaque in honor of your visit today as a distinguished scientist in the cardiovascular research program.
Okay, can everybody hear me at the back? Yes? No? Yes? Can you hear me? Yeah? Okay. Thank you. Well, it's a pleasure to be here, and I want to thank Tammy and the organizers for this great event. I've never been to Guelph before. I've driven past it many times. It's a wonderful campus, and I sort of kicking myself that I missed it all these years because my son's at Waterloo, so I just zip by. So from now on, I will stop here. The other thing is really a pleasure to see how the development has taken place here, because I remember when John and Glenn joined here many years ago, I think I wouldn't have thought that it would have been this type of intensive effort that would have materialized in such a short amount of time. And I think it's a lot of credit to the organizers.
Sorry, we're just going to increase your volume on
Go ahead.
microphone.
So where is that now? Somewhere
I
here?
think it's that one, yeah.
Okay. I don't have my glasses, so I don't see too well. Yeah.
Sorry.
Are we good?
Yeah.
No? Yes? It's good?
Yep.
Yeah? Maximum?
Yep.
Okay. Yeah, so it's fantastic to see how the build-up's gone from a molecule all the way to not just large animals, but man at Guelph. So it's fantastic to see this effort. And it's a pleasure to be a part of it. I'm hoping that we can contribute from Ottawa, not Toronto, as we move along in the future in terms of the initiative that Rob was talking about earlier. So basically, I am interested in Peter Bax, and that's why I work on the heart. So why not? Why not? And so I'm interested in how cells make decisions. And the main decisions that any cell is capable of making is to either grow, proliferate, survive, or die, or differentiate. So those are key phenomena. And I think the heart cell is unique in a way because shortly after birth, as you know, cardiomyocytes are terminally differentiated. And as a consequence, what you have is permanent withdrawal from the cell cycle, and you have a number of divisions that take place shortly after birth, and you have the formation of the heart. And after a week or so, after birth, the growth that you'll get is either physiological or pathological, depending on signals that the heart receives. Now, we know that physiological hypertrophy is driven through various programs, signaling programs, and we also know that pathological hypertrophy is driven through various signaling programs. And I guess a lot of information has been generated in trying to decipher these two arms. And I think we're still at a loss, really, to try and convert physiological hypertrophy in a way and take it away from the pathological side. There's a lot of effort in this, but it still hasn't paid off. So I'm interested in how postnatal heart actually grows. And in particular, I'm interested in what happens shortly after birth when the cells are still able to divide. And the idea being that if we can manage that, can we make more cardiomyocytes at that point, shortly after birth, to refurbish the heart? And so that's really the interest. And a lot of nice work's been done on regulating the cell cycle with the idea to entice these cells to make more myocardium. And so with that in mind, we've been interested in how this particular family of transcription factors called the E2Fs, which are present in every cell and are involved in regulating differentiation, proliferation, and apoptosis, and how they actually guide this cardiomyocyte through the cell cycle. So now, upon repeating a sort of growth signals, what happens is a series of kinases are activated, which then phosphorylate RB, and phosphorylated RB is basically removed from the complex, and E2F is able to dimerize with DP, sit on promoters of genes, and regulate these fine processes. And depending on what signals are being received, you have a set of genes turned on, which will then guide the cell through the cell cycle or to terminal differentiation. Now, the E2Fs comprise a very unique family of proteins, and there are at least 9 members that have been sort of discovered over the years. And E2Fs 1 to 5 are thought to be activator E2Fs, and the classical E2Fs, and E2Fs 6 to 8 are newly discovered in a sense compared with these, and are thought to be atypical. So what's common amongst this family is that they all have a DNA binding domain, and they sit on these genes that are responsible for these processes in every cell type. And E2Fs 1 to 5 have a transactivation domain, which can recruit RB and related transcriptional machinery in response, as indicated, to stimuli and activate transcription. However, when RB is not phosphorylated, these basically serve to repress the genes. Now, E2F 6 and 7 and 8 do not have the transcriptional domain as seen here at the C-terminus, and they do have the DP dimerization domain, at least E2F6 does. But E2F6 has a unique repression domain at the C-terminus, but none of these 3 actually are able to activate transcription, and at least that's the dogma. And I'll show you something different as we move along here. Now, there's a lot of redundancy in the pathway, and these transcription factors are expressed in a tissue-specific fashion. E2F3 activity is critical for cardiac formation, because if you knock out E2F3, you have embryonic lethality due to hyperproliferation and leads to cardiac defects. You can knock out any of the others, nothing happens. So E2F3 is the major E2F that defines cardiac formation. Now, RB, if you knock out RB, nothing happens, but there are RB family members, P107 and P130, and if you have double knockouts of these, you get no heart formation, so embryonic lethality. So the idea then to understand how the E2F pathway might be affecting postnatal cardiac development has been an issue because of this phenomena. Now, in cell culture, you can express these E2Fs up and down and knock them out and so on and so forth, and you do affect hypertrophy, apoptosis, and so on in neonatal cardiomyocyte, but you're not really learning much about the pathway in postnatal cardiac development. In human studies, it's been shown that E2F1 and RB are upregulated, suggesting that maybe they play some kind of a role in the human condition as well. So we were interested in E2F6, which I indicated is a repressor. And E2F6 is thought to recruit a repression complex, which consists of polycomb group of proteins together with MAX and MEGA, and is thought to sit on promoters and repress genes. Okay, so that's the dogma about this particular E2F member, and is thought to be activated at the E2F at the G1S phase of the cell cycle. And in cancer cells, when you overexpress E2F6, it can stall those cells and sort of prolong the S phase. Okay, so we reasoned that if we were to generate a mouse model using E2F6 in the postnatal heart, that we might be able to learn something about the E2F pathway and potentially the cell cycle shortly after birth when the myocardial cells are still able to divide. So we generated a mouse model where we express E2F6 cDNA using the alpha-myosin heavy chain promoter, with the view that since the myocardium can still divide after birth, shortly after birth, And this is a repressor of the cell cycle that we should be able to learn something about what's happening shortly after birth and maybe learn something about hypertrophy, growth, death, and differentiation. So if you look at E2F6, normal expression, at embryonic stages it's highly expressed. At P1, shortly after birth, it starts to go down. And in the adult, normally it's at very low, it's there, but it's very low levels in terms of protein. So what we did was we basically ramped up the expression at P1. And when we do that, what happens is, depending on how many copies of the gene that we titer in, we have a dose-dependent effect on death of these mice. So if we have one copy of the transgene being overexpressed, we see some of the mice dying at about 150 days of age. And if we ramp up the number of copies, they die sooner. And if we have, for example, shown here, they all have a dilated cardiomyopathy. So overexpression of E2F6 in the postnatal heart leads to a dilated cardiomyopathy that is dose dependent. And you can see that there's reprogramming going on in terms of fetal gene activation in the adult when E2F6 is being expressed. Okay? So E2F6 then, we can titrate it in, and the mice do survive, and we can study them, but they get a DCM. Okay? So the DCM actually appears as early as 2 weeks, and it sort of persists and it gets worse as time goes on, obviously. But what was interesting was, and there's sort of deficits in cardiac function as shown here, but what was interesting was there was no significant induction in hypertrophy. So we're getting a DCM right off the bat, but we're not getting any hypertrophy. Okay? So this was supposed to be a repressor. So it represses genes. Okay? That's the dogma. So Jennifer Major, who was a graduate student working in my lab, did this experiment with the microarray, and she came running into the lab and she said, look, it's not a repressor. It's actually an activator of genes. It's activating 900 genes, and only 100 are downregulated. And then she said, the genes that are getting activated are all E2F responsive. Okay? So it's contrary to what the literature was saying. And so she was saying, well, you know, I don't think I'm going to get my PhD now. This is all different. I said, no. And I said, this is really exciting, you know. I said, this is a repressor in different types of cells, but in the heart, nobody's really examined it. It's actually an activator. So the genes that it was activating are involved in DNA replication, DNA repair, cell cycle, and it actually is regulating the other E2Fs in terms of their mRNA expression at least. So to me, that was very exciting. And so then we started thinking that maybe this could be, since it's a major regulator and a master regulator, that maybe this could be a target, and in fact, treating cardiac disease in the adult, because the cardiomyocytes terminally, they drove from the cell cycle shortly after birth, and we are upregulating this at birth. So if we upregulate it later, maybe we won't have the DCM, and maybe we have all this good stuff going on. Okay, so that's the way I was thinking. Anyways, more to what she did. So Jennifer looked at the E2Fs, and she found that E2F3, which is the major regulator of transcriptional control mechanisms in the heart, was completely downregulated by E2F6. So in the transgenic mouse, E2F3 is there, but it's much depressed. And she also found that E2F6 was present at promoters, which are being activated. Okay? So it's working as a transcriptional activator. So she came up with this model that when you have normal levels of E2F3, you have repression of genes. And then when E2F6 is present, what it's doing is it's altering E2F3. It's taking it away from this complex. It's substituting itself because it can bind to DP because it has the DP binding site, and somehow is able to recruit a transcriptional complex to activate all these E2F responsive genes, because E2F6 can bind to all these promoters. So when she looked at that, she became very excited, because now she has a real PhD project. So then she looked at what happens to E2F1 which has been worked on extensively in the heart. And it turns out, even though the mRNA was upregulated in the transgenic, as I showed you in the previous slide, the actual protein doesn't actually change much. The only E2F protein that really changes is E2F3. It goes down quite significantly. And what was really intriguing was that she found RB was also downregulated in the transgenic mouse. So if there's less RB, then you're going to get more transcription, okay? So it was all pointing to E2F6 having a major regulatory role in terms of upregulating gene expression programs as opposed to repressing. So DP, so since E2Fs need DP, so what's happening to DP in these hearts? And sure enough, in these young pups, she found that DP is upregulated by E2F6. So in the transgenic mouse, you have a huge upregulation. So there's DP being produced in these hearts so that it can bind to, and here's the binding experiments. It's an IP of DP and E2F6, and they interact. So E2F6 upregulates and binds DP to sit on those promoters and somehow activate transcription. So since these mice show induction of DNA repair and all the good genes, we reasoned that maybe E2F6 was anti-apoptotic, was actually a survival factor, okay? So she looked at this and she found that really there was no apoptosis in these transgenic pups that were starting to develop DCM. She did see a little bit of apoptosis in adults, but wasn't very significant. So there's no real change in apoptosis in these hearts that are getting a DCM. So it's not due to apoptosis that we're seeing the phenotype. In fact, when she looked at the survival pathway, it was upregulated in these pups from these transgenic hearts, as shown here. And the major survivor, as you know, survival protein is BCL2, and you can see it's massively upregulated. So the entire pathway, phospho-SOC, ERK, and AKT are all upregulated, which lead to survival of these cells. So it's not, the DCM is not due to cell death. Okay? So she tested this further. She know that cobalt chloride can basically induce cell death of cells when you incubate cells in this particular compound, and basically it's thought to be due to activation of the HIF pathway, and that's been worked out in cells. And so she used this in cell culture of cells that were isolated from transgenic hearts versus wild type. And doxorubicin, as you know, can induce the DCM due to DNA damage. And since E2F6, as I've indicated, is activating repair genes, we reasoned that maybe it was anti-apoptotic and may actually attenuate the death pathway. And so this is one experiment she did where she looked at cobalt chloride treatment on isolated cardiomyocyte from wild type and transgenic hearts, and she found that there was more of a survival of the transgenic cardiomyocyte versus the wild type. And then once you look at the biochemistry, you could see that in the wild type you have activation of the caspase pathway, whereas in the transgenic it's attenuated. So basically E2F6 is working to actually save the cardiomyocyte against death, although we're getting a DCM. Okay? So what about the cell cycle? Well, when you look at the transgenic heart and do the analysis, what you find is cyclin B is downregulated, shown here, and you find that cyclin E is upregulated, shown here. So that means that the cells go into they're enhanced to go into the S phase, but they get stuck here for a while. So you have a change in the cell cycle, which we had predicted would happen because we thought it was a repressor, but it turns out that in the S phase, when you prolong the S phase, you are actually stimulating genes that are involved in DNA repair and DNA synthesis and so on and so forth. So this particular repressor is actually stalling the cells, cardiac cells, in the S phase, allowing the profile of genes that I've indicated. And there's no change in BID incorporation, so there's no change in proliferation, and there was no change in hypertrophy. So no hypertrophy, no proliferation, no cell death, but we're getting a DCM, okay? An upregulation of E2F responsive genes. So she looked at the data again, and she came very excited. One day she said the number one change that she found in the microarrays was the ketogenic enzyme beta-hydroxybutyrate dehydrogenase. It was screaming hot. And as she was looking at this, a couple of papers came out in Circ Research where they'd indicated that BDH1 may be a biomarker in human heart failure, and that this may be a critical switch in terms of substrate that happens during cardiac disease. And so when she looked at her data, she found that the BDH1 transcript levels were up about almost 60-fold. And when she looked at the protein profile, this was very intriguing. So here's the wild type. BDH1 in the wild type heart doesn't come up till 6 weeks. There's nothing at P1. In a transgenic, she had at P1 as much BDH expression as you do in the wild type. So there's a complete switch at day one in these transgenic hearts to the ketogenic enzyme BDH1. So what's going on? Very interesting switch. Again, it's an upregulation of a gene due to a repressor, okay? So which was, to me, was still, I was still trying to get over that. Anyway, so does E2F6 activate a metabolic switch? And ketolysis is activated when glucose is scarce. And we know that in the neonatal and the postnatal myocardium, glucose is the main energy source, right? In the embryonic heart and the neonatal heart. So are we getting a switch in metabolism due to the stalling of the cell cycle, which is then impacting ketolysis, which then might be impacting the DCM? Because we're getting a DCM, remember that, right? We're getting all the good things happening, no hypertrophy, no cell death, but we're getting a DCM, okay? So then she started to look at this more carefully. So she isolated the cardiomyocytes from the wild type and the transgenic hearts, did the Seahorse analysis, and found that indeed glycolysis was down. Indeed, GLUT4 was down in the transgenic, as was the pathway involved in glycolysis. And sure enough, we're affecting the metabolic pathway in these cardiomyocytes, which is leading to a switch in metabolism, and that then is somehow leading to a DCM. So what do ketones do? Because BDH1 is involved in ketolysis. So what do ketones do? So she looked it up, and she found that ketones are actually pretty famous molecules involved in signaling, and they have their own receptors. At least beta-hydroxybutyrate has its own receptors that it can bind to, and that can actually lead to changes in second messengers as well as changes in gene expression patterns. Beta-hydroxybutyrate can actually impact the epigenome but through histone deacetylases, and it can impact the metabolism in terms of substrates and generate acetyl-CoA, which can also impact acetylation. So basically, this is a very important pathway, both through signal transduction as well as going directly to the nucleus to regulate the epigenome, and therefore can impact major phenomena. So how then this might be leading to a DCM was the question. So she started to look at the various aspects of this phenomenon. And what she found was that Titin cap was markedly downregulated, as shown here in these hearts, as was connexin 43. So connexin 43, which is the major gap junction, as you know, which is involved in communication from one cell to the next, was downregulated. Connexin 43 wasn't affected, and Titin cap was downregulated, okay? And so here's the ECG showing that, yes, we were getting sudden death, probably due to an arrhythmia, maybe due to atrial fibrillation. We didn't examine that, Peter, but I think we should. Now, a paper had come out just about then showing that in endothelial cells, beta-hydroxybutyrate can upregulate connexin 43 expression. So she performed that experiment in wild-type cardiomyocytes, and sure enough, beta-hydroxybutyrate can upregulate connexin 43 expression as shown here. And we got very excited and we said, let's see what happens in the transgenic hearts, where we showed that in vivo connexin 43 was down, right? I just showed you that data. But when she plated these cells and treated these cells with beta-hydroxybutyrate, what she found was that connexin 43 was already upregulated as she plated them. So when she added the beta-hydroxybutyrate, it had no effect. So I don't know what's going on in terms of culture conditions. So cell culture sometimes doesn't behave as you think it should. And so therefore, she wasn't as excited when she saw this result, and she said, well, can we not mention that? I said, no, that has to go in because it's contrary to the hypothesis. But we just don't know why the in vitro data here in terms of connexin 43 is different from the in vivo data here as we look at the heart. So anyway, so weren't able to really prove our hypothesis here, but she did an immense amount of work on this, and she took this project from basically generating the transgenic mouse with one of the postdocs, and actually found this entire mechanism through E2F6. And what was really, and she had to leave because she got a CIH postdoc fellowship. She's in Denver, Colorado, in a big lab doing some great stuff. But she still phones and says, I still want to know how this repressor is working to upregulate the gene expression program, And so I have another student working on that, and I'm going to tell you about it because I don't want to spoil it for the student because they're still trying to figure this out. But it turns out that it may be linking in with the hippo pathway, which also regulates cell growth, death, and differentiation. And in fact, when we cloned E2F6, we did it through a screen, through a protein that I discovered called SLMAP. And SLMAP is a now key player in the hippo pathway, and it's linking into this. And so as far as this project goes, so we're trying to work out that aspect out. And we're also trying to work out whether in human cases we have changes in the E2FRB pathway, particularly in idiopathic DCM. There's about 1/3 human cases where we don't know why they get a DCM. And certainly talking to Glenn yesterday, that he's working on DCM in the dogs, it might be worth looking at this pathway, Glenn, to see if there are some changes that might be leading to this. And clearly BDH1 may be a player in terms of metabolic switch, not just in heart failure, but in DCM as well. And so we're trying to look into that further. And what my favorite project right now is actually use gene therapy with E2F6 in large animals, hopefully, to see that if we can protect the adult heart using E2F6 against hypertrophy and cell death due to insult, because those are the major things that happen when you have an MI. Okay? And so that's where we're going with this. And finally, I just want to thank Jennifer Major, who did about 90% of this work. And she published 3 papers as first author, and she was highly rated at the CIHR, and she deserved to be. Okay? And Bart Westendorp, who was the postdoc who was working on the project with Jennifer, at least some of it, he's a professor now at Utrecht in Holland, and very talented young man as well. And the rest of the crowd is still trying to graduate. Okay? Thank you very much. Sure, go ahead.
Just a quick question with regards to your E2F6 construct. It's MIC tagged at the N-terminus.
Right.
And MIC is slightly acidic. The
Yeah.
MIC tag is slightly acidic.
Right.
Is there any possibility that that could be functioning as a transcriptional activation domain, even though it's fused at the N-terminus of your DNA binding domain?
Yeah, that's a good question. We use that in 2 other genes that we're working on, which are related. It doesn't happen, so we don't think it's the MIC. But it's a good question. Yeah. Yeah, Peter?
So you point out that E2F6
6.
the cell cycle, right? Controlling cell cycle and controlling checkpoints.
Right.
So did you actually look to see if there's differences in like polyploidy or the number of nuclei and whether or not that's different in development?
Yeah, well, that's a great question. So E2F7 and 8 have been shown to have an influence on that, but E2F6 doesn't. So we did look at that. I don't know how thoroughly we looked at that time, but we didn't see a change in those parameters. But E2F6, 7 and 8 have certainly been linked to that, but in different systems.